So students, welcome back after this break. So if you have any questions, is everything we discussed in the last class, is it clear? Any question, any query?
Oh, yes, sir.
Okay, everything clear?
Am I audible?
Yes, Shakti, you are audible. Any question?
Sir, is there any role of the framework region?
See, definitely it has a role, but not that important role, right? Because see, the variability is also there in the framework region, right? It's not constant among all the different types of antibody, right? So obviously to find a particular epitope, right? These hypervariable regions are important, right? But the amino acids in the framework region, right, they will also somewhat help in binding of the epitope, right? So they definitely have a role, but not that important role as if you compare it with the hypervariable region. Okay, so it's basically a frame. ठीक है, अगर तुम्हें शीशा टांगना है, so you have to put it in a frame, right? So उसको इस तरह से imagine करो, then it would be clear that this is your frame, and in this frame there are hypervariable regions, right? So that becomes your framework region and the hypervariable regions. Okay, so if you see it in this way, it looks easier to understand. Okay.
Yes, sir. Thank you, sir.
Okay, any other question? Okay, then let me start with today's class, and this is your like the practical topic. Okay, so if you remember the last time we uh, had this practical, it was of serum separation, right? So in that class we discussed about the difference between serum and plasma, right? And we also talked about how we take out blood, right? How we store the blood, and um, I remember we also talked about the different types of vials that are available in the market, right? So depending on what kind of test you want to perform, you can put the blood in different types of tubes, and the difference is that those tubes contain some reagent, right? It could be heparin or some other agent, right? So which helps in coagulation of the blood, or for whatever purpose it is being added. Okay, and I think we also talked about vacuotenors, right? So I hope you now know how we take out blood from a person. Okay, and how we separate the components, right? So basically, for any biochemical test, you would require serum, okay? Uh, and for some of the test, you would require whole blood, okay? So, like suppose you want to study some component of RBC, okay, then you will not separate the serum, because then what happens, there is a formation of clump of those red blood cells, right, which we don't want. So depending on what kind of test you need, okay, uh, there are some tests in which hemolysis, we want to see hemolysis, okay. So again, for those kind of tests you will require the whole blood, okay. So what happens, you just take out the blood, okay. So you can keep in one vial, you can keep the whole blood, okay. In another you can put the reagent so that you get the serum, okay. So you can just separate the blood on your requirement, okay. Now today we are going to talk about ABO blood grouping, okay. uh, so do you know why we carry out ABO blood grouping? So students, just raise your hands, switch on your camera, and then tell me the answer. Why do we uh, carry out this ABO blood grouping test? I'm sure all of you know this. Okay, okay. So I can see three hands being raised. Manaswini, Sanjana, any other person? Okay, Sam. Okay. So let me bring all of you together. So just switch on your cameras. I'll put you in the group mode. Switch on your cameras. I'll put you on the same table. Okay. So I'll start with Manaswini. She raised her hand first. So Manaswini, why do we carry out blood grouping?
Sir, so let's say there's a patient who is in need of blood. Then let's say the extra blood that you're giving them, it has to be of the same blood group. So, or it has to be from a donor which can donate blood to a different blood group. So, because of that, knowing the ABO blood grouping, I mean performing that will be very important in the case of blood transfusion or even organ transplantation.
Okay. So basically, if there is some trauma, there is some need for giving blood to a patient. So for that purpose, we need to have this information, right? Okay. So Sam and Sanjana, what are your points?
So basically, like if someone
So,
with
okay,
yeah, go on.
Sam, you go.
Yes, Sam, you are not audible. Sanjana, Sam is not audible. What is your point?
Am I audible now?
Yes, you are audible, Sam.
Yes, sir. So blood has different antigens and antibodies. So when someone with A blood group is introduced with B, there is high chance of the immune system attacking the new blood.
Okay, okay, fine. So Sanjana, where is the antigen and where is the antibody? Do you know this?
Sir, so the red blood cells have the antigen on their surfaces.
Right,
So if the the blood contains the red blood cells with, for example, type A antigen on their surface, and it has antibodies against type B red cells. So if a
okay,
transfusion takes place, so type B will be so antibodies will be produced against the
right,
recipients.
right. I got your point. So basically, uh, see, it's all about antigen and antibody, right? So, uh, so, uh, okay. So when we discuss ABO blood grouping, you need to understand that individuals have different kind of blood group, okay, and I'm sure you have read in your school also how that blood group is determined. So it's the genetic level, right, which determines what kind of blood group you have, okay. So the blood cells, they have a specific antigen on their surface, right, and when you are growing, right, so your immune system has seen those antigens, okay. So the blood which I am having, my immune system sees that blood as self, okay. My immune system recognizes those antigens which are present on my blood as self, okay. But if I get blood from some other person who is having some different blood type, right, then our immune system will start attacking that blood, okay, and it will lyse all the blood which has entered, and that can create a lot of problems, okay. I'm not going to discuss that right now because the issues related to blood transfusion we will read in hypersensitivity, okay. So at that time we will discuss what are the symptoms when wrong blood is transfused in a person, okay. What happens if wrong blood is transfused in an individual, so we will understand at that time, okay. Now the ABO blood grouping test, okay, it is based on agglutination reaction, okay. The simple test, it's based on agglutination reaction. It gives you the result within minutes, okay. And the principle is basically the interaction of antigen and antibody, okay. So again, we in this level, we are not going to discuss antigen antibody interaction in detail, right? But antigen antibody interaction is one of the most important factor for diagnostic tests, right? Uh, okay, so when we talk about agglutination, so basically what happens, these antigen and antibody, they bind, and because they bind, they form visible clumps, right? So if I put blood, right, or if I put antibodies on a surface, right, they are not visible to our naked eyes, right? But when they agglutinate, when they clump, so we can see those clumps with our naked eyes, okay? And that is the principle of ABO blood grouping test. So what I'll do is I'll show you a video of ABO blood grouping. Okay, it will also show you how we perform this test, and after we see this video, we'll have more discussion on this test, right? And if we get a chance, so if the university opens and if we get a chance to uh, work in the laboratory, so then we will carry out this test, okay. So it's a very simple test. It gives result within minutes. Okay. So let's first understand how this is done. So let me share the video.
In this video lecture we will understand in detail A. VO blood group antigens, A. VO blood group antibodies, and we will also find out from where A. VO antibodies came. The blood type of a person is determined by the antigens expressed on the red blood cells. In 1990s, an Austrian scientist, Karl Landsteiner, discovered a VO blood group system. Under this blood group system, human blood is grouped into 4 principal types, designated as A, B, and O. Let's first understand A, VO blood group antigens. The blood group under this system is determined by presence and absence of the 2 antigens on the cell membrane of RBCs, namely A and B. These antigens are carbohydrates. Presence or absence of these antigens depends on individual's DNA. The DNA express different types of enzymes. These enzymes catalyze a series of reactions in which they transfer sugar units. These enzymes are known as transferases. These enzymes are responsible for the type of antigen expressed on red blood cells. Let's understand this by illustration. Let's say this is a red blood cell. A single RBC has about 2 million blood group antigens on its cell membrane. But, for our illustration, we will take only 2. All blood type individuals express a precursor oligosaccharide on their RBCs. So, this is a precursor oligosaccharide. All humans also express a transferase enzyme that transfers and attaches fucus to this precursor oligosaccharide. Binding of fucus to the precursor oligosaccharide results in the formation of the H antigen. Now let's see what is the difference between antigens present in different blood types. The DNA of blood group A individuals express an enzyme which attaches N-acetylgalactosamine to the H antigen. With the attachment of N-acetylgalactosamine, carbohydrate chain now becomes antigen A. In individuals having blood group B, a different enzyme attaches galactose to the H antigen. With the attachment of galactose, antigen B is formed. In individuals with A, B blood group, both enzymes, that is, one that transfers N-acetylgalactosamine and second, that transfers galactose, are expressed. Therefore, RBCs of these individuals express both A and B antigens. The individuals having O blood group have only H antigens on their RBCs. These individuals do not have enzymes that transfer N-acetylglucosamine and galactose to H antigens. Therefore, neither A nor B antigens are expressed by the RBCs of blood group O individuals. Let us now study the A-VO blood group antibodies. Now question is how to determine which type of antibody will be present in a particular blood type. To know the answer, always remember this: our immune system forms antibodies against the missing A-VO blood group antigens. Or in other words, antibodies are found against the antigens which are absent on the cell membrane of RBCs of an individual. Let's understand this in detail. In an individual with blood group A, antigen B is missing. Therefore, antibodies against B antigens will be found. These antibodies are known as anti-B antibodies. Similarly, an individual with blood group B will have anti-A antibodies. This is because here antigen A is missing. Individuals having A, B blood group have both the antigens, that is, antigen A and antigen B, present on their RBCs. No antibodies will be found in these individuals because none of the antigens is missing. In blood group O individuals, both antigen A and antigen B are missing. Hence, both anti-A and anti-B antibodies will be found. Let us now recall what we have understood in this video lecture. There are 4 blood groups in humans. These are A, B, and O. Blood type A individuals express antigen A on their RBCs and anti-B antibodies are found in their serum. Blood type B individuals have antigen B and anti-A antibodies. Blood type A, B individuals have both antigen A and antigen B. They do not have any antibodies in their serum. And blood type O individuals do not express any antigens. They have both anti-A and anti-B antibodies in their serum. Now we all know that antibodies are formed when our immune system is exposed to non-self antigens. Here we saw that antibodies are present against the antigen which is absent or missing on the cell membranes of RBCs. Now the question here is, from where these antibodies against the missing antigen came? Let's understand this concept. Antigens similar to A, B, O blood group antigens are found in nature. They are expressed by common microorganisms, those found in human intestines and in many foodstuffs. We are exposed to these antigens early in the development of our immune system. Let's say these are bacterial cells having antigen A and antigen B similar to human blood group antigens on their surface. Now suppose an individual has blood group A. This means the RBCs of this individual express antigen A on the cell membrane. This individual is exposed to similar antigen A and also to antigen B of the microbial surface. Since antigen A is already present, it is recognized by the body as self-antigen. The similar antigen on microbial surface, that is antigen A, is also recognized as self, and no anti-A antibodies are produced. But these microbes also have antigen B. The A-type individual's red blood cells do not express antigen B here. That is, antigen B is absent or missing. This microbial antigen B is recognized as non-self. Therefore, anti-B antibodies are produced against the microbial antigen B. Thus, in an individual with blood type A, anti-B antibodies are found in the serum. So, now we understand that antibodies against the missing A, B, O blood group antigens is present in an individual.
So student, is it clear? Okay, let me complete this video. Okay, so students, any questions? Okay, I think we can have attendance, and after attendance we will see exactly how we carry out this test in lab. Okay, so students, be ready with your cameras. Adish, you are not visible.
Present, sir.
Present, sir.
So am I visible?
No, Adish.
Present, sir.
Yeah. Atoiba. Atoiba, you are not visible. Ayushi,
Present, sir.
Sam,
Present, sir.
Devanshi,
Present, sir.
Devanshi, you are not visible. Okay, Ishita, Janvi,
Sir, I'm present.
okay, okay. Janvi, Kshitij,
Present, sir.
Manaswani,
Present, sir.
Namrita,
Present, sir.
Nandini,
Present, sir.
Nehal, Rupal, Rupal, okay, Saiba, Sahiba, Sanjana,
Present, sir.
Shakti,
Present, sir.
Shivani, Tanvi,
Present, sir.
Tridiv,
Present, sir.
Vivek, Yukta,
Present, sir.
Yukta, you are not visible, and Yuvraj, Yuvraj, Janvi. Okay, students, so let's now see the practical aspect also, okay, how we determine the blood group. So again, I will show you a video on this.
Hello everyone. In this segment of the biochemistry, we are going to be discussing about the procedures involved in identifying the blood types. Aim. The aim is to identify the type of the blood that a person has. Blood typing is done so that you can safely donate your blood or receive a blood transfusion. It is also done to see if you have Rh factor on the surface of the red blood cells. Blood is often grouped according to the Landsteiner's ABO blood grouping system, and the four major blood types according to this classification includes type A, type B, type AB, and type O. The A and B are antigens which are commonly detected on the surface of the red blood cells. Materials required: freshly collected sample blood, porcelain spot plate, normal saline, a small test tube, a dropper, and anti-sera of anti-A, anti-B, and anti-D. The procedure of blood typing: first, fill half of the test tube with normal saline. Add 2 to 3 drops of blood to the test tube containing the normal saline. Now take clean porcelain spot plate and then mark them A, B, C, and D. Put 1 drop of anti-sera solution, anti-A, anti-B, anti-D in A, B, and D porcelain spot plate respectively. With the help of a dropper, add drops of normal saline mixed with blood in all of the spot plates. Now gently shake the porcelain spot plate and wait for 10 minutes. Look for agglutination and compare with the control C. If dust particles like appearance is visible, then it is agglutination. Now inferences after observation. If agglutination is present in spot A, then the blood group has A antigen. If agglutination is present in spot B, then blood group has B antigen. If agglutination is present in spot D, then the blood group is Rh positive. If agglutination is absent in spot D, then the blood group is Rh negative. If agglutination is present in both spot A and spot B, it means that the patient has both A and B antigens on the surface of his RBCs. Now here's an image where you can see agglutination in A, B, and D spots in the porcelain spot plate, which then gives the blood group as A, B, positive. The positive denotes Rh positive.
So students, I'm sure it's clear.
Yes, sir.
Any question? So I'm trying to show you another video which actually show you the real lab conditions, because you've just seen the animation right now. So till that time, if you have any question, you can ask. I'm just trying to find the video because you should know exactly how that clumping look like. Okay, so let me share the video.
Solution. In this video, today we will study about blood group determination of your own sample. First of
So this test is bit different from what we saw in the animated video, right? In that we took the blood from a test tube, right? Or from that vacutainer, right? So what happens? Suppose you have taken out blood for suppose lot of different tests, right? So the technician, they will just take the blood, right, and from that blood they will use for ABO blood grouping, right? But if you have just one test to do, if you just need to do this blood grouping test, then you don't need to take out the blood from the vein, arm vein. Okay, so you can just prick the tip of your finger, okay, and just carry out this test. So just see how it is being done.
all, sterilize your little finger with the help of ethanol, then adjust the depth of pricker paint
So
and
the,
prick it.
so the first thing was to clean your fingertip, okay, because obviously you'll have to prick a needle in the fingertip. So first clean your fingertip with the help of ethanol or spirit, whatever you have, and then you just need to prick the needle into your fingertip.
To your finger. Allow a drop of blood to come outside the finger and place the 3 drops of blood respectively in the position marked as A, B, and D. For your convenience, we have replaced D with a plus or minus sign. It will be easy to understand the ultimate last result. After placing 3 drops of blood on
Okay, so you can see three drops of blood have been put on a slide, right? So in the animated video, you saw that porcelain whatever plate or whatever it was. Okay, but here you are seeing that we can also use this glass slide. Okay, and this glass slide is easily available in the laboratories, so you can just use it, and then you can see three bottles, right? So one, on one it is written A. Okay, this A has anti-A monoclonal antibody. Okay, so which means it has antibodies against the A antigen. Okay, and it's a monoclonal antibody. So we will talk about monoclonal antibodies in once we cover antibodies. Okay, here you can see this is B. This solution, it contains anti-B monoclonal antibodies, right? So antibodies against the B antigen. Okay, and this is anti-D or antibodies against RH antigen. Okay, so now what you need to do is you need to put a drop of this, right, on the blood drops and then mix it.
On their respective position, place one drop of antiserum A to
Okay, students, another thing, never touch this solution with the blood. Okay, because then you will contaminating this solution. So always you have to put the drop from above, right? Or you can put the drop like side by side. Okay, uh, without touching the blood drop. Okay, so I'll just go back and show you again.
place one drop of antiserum A to A drop of blood. Same, place antiserum B to the B drop of blood. And at the last, place D antiserum in place of plus or minus sign. Please remember we have replaced the D in place of plus or minus. Once we have added all antiserum to their respective drop of blood, mix thoroughly with.
Okay, so as I said, once you have put the drops, you have to mix it. Okay, and for mixing, like in this case, he has taken toothpick, right? So we can use sterile toothpick or we can use glass tubes, right? So whatever you take, but it should be clean, it should be sterile, and you just need to mix it. Okay.
The unused matchbox stick or toothpick. Kindly do not use the same toothpick for mixing purpose of another. Use one for one mixing. We have mixed all three drop of blood with their.
Okay, so you need at least three toothpicks. Okay. सो जस्ट डोंट थिंक कि हमने क्योंकि तीन अलग अलग सॉल्यूशन लिए हैं सो वी कैन जस्ट डू इट विद वन टूथपिक नो बिकॉज इफ यू डू दिस अगर बी में सपोज यू फर्स्ट डिड इट इन ए so you will be picking those antigen and putting in B, right? So you don't have to mix it. So you have to use three different toothpicks for this uh, purpose. Okay.
Respective antiserum. Now wait for few minutes. There will be a coagulation or agglutination process. As you can see on the screen, there is easily an.
So students, you can see clumping in B, right? You can see the clumping in RH, right? But you cannot see any clumping in this A. So this is B positive. Okay, the blood group in this case, in this example, is B positive because you can see the clumping in both B and RH.
Clearly visualize the agglutination of B as well as plus and minus sign. It indicates the blood is belong to B and positive, so our blood group is B positive. The agglutination process is very much clear. So at the last.
Okay, clear? I can again show you another video.
Procedure. Swab the finger with cotton balls dipped in a spirit and take blood sample using a lancet as shown. Take 3 separate drops of blood on a glass slide. Place a cotton ball on the finger to stop the blood flow. Pour 1 drop each of anti-A, anti-B and monoclonal D on each drop of blood respectively. Mix the monoclonal antibodies with the blood sample properly using a toothpick. Make sure you take a new toothpick for mixing each drop. When RBCs carrying one or both antigens are exposed to the corresponding antibodies, they agglutinate, that is, they clump together. For example, in this slide, agglutination occurred on the first and the third drops, which corresponds to antigen A and Rh factor.
So students, which would be this blood group?
A positive.
A positive.
Yes, so you can see clumping here and not this. So this is A positive, okay? So clear? So I hope ABO blood grouping is clear. Any questions? So students, this is the second practical that we have done. So I have not received any written practical from your side. So now we have done 2 practicals, right? So you will have to prepare that practical file. So prepare it and send these 2 practicals to me. Okay? So any question on ABO blood grouping?
Sir,
Yes, Shakti.
so suppose we don't get any clumping in both A and B and we get it in D, so it will be O positive.
Yes.
Okay, sir.
And suppose you don't see clumping in any of the 3 drops, then?
O negative.
Okay. Okay, and yes, so we are not just looking for the A and the B antigen, right? We are also looking for the Rh factor, right? And again, Rh factor is very important, right? We will talk about one of the disease condition in children, right? In which this Rh factor plays a very important role. The disease is called erythroblastosis fetalis, right? It happens in females who are Rh negative, but they are carrying a baby which is Rh positive, right? So what happens? The female body, uh, obviously the immune system has not seen that Rh antigen, but the baby which is inside that woman, that baby is having the Rh positive blood group, right? So in that case, the immune system will start attacking the baby. Okay, it will start attacking the fetus, and that can lead to death of the unborn child, right? So that is one of the disease condition. We will talk about this in hypersensitivity. Okay. Okay, so students, if no more questions, then I am going to end the class now. Is it okay?
Yes, sir.
Okay, so I'm ending the class now. I'm stopping the recording.